Welcome to our exploration of oogenesis, the remarkable process of female gamete formation. Oogenesis is the process of female gamete formation that occurs in the ovaries. Unlike spermatogenesis in males, which produces millions of sperm continuously, oogenesis produces a limited number of eggs during a female's lifetime. The contrast between these processes is striking. While males produce billions of sperm continuously from puberty through adulthood, females are born with all the eggs they will ever have. The process of oogenesis spans from fetal development through a woman's reproductive years. It begins during fetal development with primordial germ cells, continues with Oogonia multiplication between 8 and 20 weeks, and by birth, primary oocytes are formed and arrested in prophase 1 of meiosis. At puberty, monthly egg maturation begins, continuing through the reproductive years until menopause, when viable follicles are depleted. The cellular journey of oogenesis begins with primordial germ cells that differentiate into Oogonia. These Oogonia multiply through mitosis, creating millions of potential egg cells. The Oogonia then develop into primary oocytes that enter the first meiotic division but remain arrested in prophase 1 until puberty. To summarize, oogenesis has several unique characteristics that distinguish it from male gamete formation. Unlike spermatogenesis, oogenesis produces a limited number of gametes, all formed during fetal development. These primary oocytes remain in meiotic arrest from fetal life until they're activated during puberty. During each menstrual cycle, typically only one functional ovum is produced. The process results in large, nutrient-rich egg cells capable of supporting early embryonic development. Now that we understand the basics of oogenesis, we'll explore follicular structure and classification in the next section. Ovarian follicles are the fundamental functional units of the ovary, each containing a developing oocyte. Let's examine the structural components of follicles. At the center of each follicle is the oocyte, containing a nucleus with genetic material. Surrounding the oocyte are granulosa cells, which provide nutrients and developmental signals. The basement membrane separates the granulosa cells from the outer theca cells. The outermost layer consists of theca cells, which produce hormones essential for follicle development. Now let's examine how follicles are classified based on their developmental stages. Follicles progress through several distinct developmental stages. Primordial follicles are the most basic stage, containing a primary oocyte surrounded by a single layer of flattened granulosa cells. As development continues, the granulosa cells become cuboidal in shape, forming a primary follicle. Secondary follicles display multiple layers of granulosa cells and the beginning of theca cell formation. The most advanced stage is the antral follicle, characterized by a fluid-filled cavity called the antrum, with the oocyte pushed to one side. These follicular stages also differ significantly in size. Primordial follicles are microscopic, measuring only about 30 micrometers in diameter. Primary follicles roughly double in size to about 60 micrometers. Secondary follicles continue to grow, reaching approximately 100 micrometers. Antral follicles are substantially larger, typically measuring 200 micrometers or more. Understanding these structural differences is essential for comprehending follicular development and function. Granulosa cells are essential, providing nutrients and growth factors to the developing oocyte. Theca cells produce androgens that granulosa cells convert into estrogens, driving follicular development. The formation of an antrum signals advanced follicular development. While many follicles begin development in each cycle, typically only one will reach full maturity and ovulation. With this understanding of follicular structure and classification, we can now explore the hormonal regulation that drives follicular development. Follicular development is precisely controlled by hormonal signals from the hypothalamic pituitary-ovarian axis. This complex system involves the hypothalamus, which releases gonadotropin-releasing hormone, or GnRH, signaling the pituitary gland. The pituitary then secretes two key gonadotropins, follicle-stimulating hormone and luteinizing hormone, which travel to the ovaries. The ovaries then produce hormones that provide feedback to the hypothalamus and pituitary, creating a regulatory loop. Follicle stimulating hormone, or FSH, plays a critical role in follicular development. FSH stimulates the growth of primordial follicles into primary follicles, initiating follicular maturation. As FSH levels increase, it promotes granulosa cell proliferation and enhances estrogen production within the developing follicle. Luteinizing hormone, or LH, becomes crucial in the later stages of follicular development. When LH levels surge, they trigger the final maturation of the dominant follicle into a Graafian follicle. This LH surge ultimately triggers ovulation, where the mature follicle ruptures and releases the oocyte. After ovulation, LH transforms the remaining follicular cells into the corpus luteum, which produces progesterone. Beyond the pituitary hormones, several locally produced factors within the ovary play crucial roles in follicular development. Inhibin is produced by granulosa cells in developing follicles and helps regulate FSH secretion through negative feedback to the pituitary. Anti-Müllerian hormone, or AMH, is another important local regulator. AMH helps regulate the rate of follicular recruitment and modulates follicle sensitivity to FSH, serving as an important biomarker of ovarian reserve. Activin enhances FSH action within the follicle and promotes granulosa cell proliferation. The complex interplay of these hormones ensures that typically only one dominant follicle reaches maturity during each menstrual cycle in humans. As one follicle begins to produce more estrogen and inhibin, it suppresses FSH levels, which prevents other follicles from continuing to develop. This dominant follicle continues to grow and ultimately undergoes ovulation, while the other follicles undergo atresia, or degeneration. In summary, follicular development is regulated by a complex interplay of hormones from the hypothalamic pituitary ovarian axis and local ovarian factors. This precise hormonal coordination ensures that typically only one dominant follicle reaches full maturity and ovulates during each menstrual cycle. The follicular development cycle follows a precise timeline throughout the menstrual cycle. During the early follicular phase, follicle-stimulating hormone recruits several primordial follicles to begin development. These follicles grow through primary, secondary, and tertiary stages, developing multiple layers of granulosa cells. Through a selection process called follicular dominance, typically only one becomes the dominant follicle, while others undergo atresia or degeneration. Just before ovulation, the oocyte within the dominant follicle resumes meiosis, which had been arrested since fetal development. Ovulation occurs when the follicle wall ruptures in response to a surge of luteinizing hormone, releasing the mature oocyte. After ovulation, the remaining follicular cells transform into the corpus luteum, which secretes progesterone to prepare the uterus for potential implantation. If pregnancy doesn't occur, the corpus luteum degenerates after about 14 days, forms a corpus albicans, and a new cycle begins with recruitment of new follicles. Throughout the menstrual cycle, follicular development follows a carefully orchestrated pattern from recruitment and selection to ovulation and corpus luteum formation.